Okay, so this is the second part in the audio lecture covering cell structures. So we'll be looking at internal structures and special structures for this audio lecture. So the first structure we'll look at is the cytoplasm. If you remember from part 1, we talked about how the cytoplasm is a structure that's going to be found in all cells. So this is a very gelatinous solution. So it is 70 to 80% water, but there's also going to be lots of proteins and other cell masses such as ribosomes and granules and actin strands that are going to be found within it. And then within the cytoplasm is where we have biochemical and synthesis reactions occurring. So the next structure we'll look at is genetic material. Again, a structure that's found in all cells. Between prokaryotes, bacteria, and eukaryotes, it's a little bit different how the genetic material is going to be packaged. And so first we'll look at bacteria. So for bacteria, they're actually going to have a single circular strand of DNA. It is double-stranded, just like with eukaryotes. And what happens, it actually aggregates together and forms nucleoids. So it does not get packaged into a nucleus like we see in a eukaryote. And so it's sitting there in the cytoplasm, just kind of aggregated together. Some bacteria also have plasmids, and so these are known as non-essential pieces of DNA. So the bacterial chromosome contains all the genes that the cell needs in order to live, that the bacteria needs in order to live. In the plasmid, it's kind of extra pieces that makes life easy, but not required. The plasmid is also double-stranded and in circles, and so some bacteria can have multiple plasmids. So there's some bacteria that have 7 to 13 plasmids in them, and they all would contain different luxury genes. So genes that might code for drug resistance or production of a toxin or an enzyme. Again, that would make their life easier, but not necessarily required for it to survive. So if you think about a bacteria on its day-to-day life, it's not going to be exposed to an antibiotic, right? We're not taking antibiotics every single day. And so the bacteria living in our gut don't need genes to be resistant to antibiotics every single day. But sometimes, maybe we get sick with something else because we were exposed to something that was opportunistic. And so this is going to allow the bacteria to survive that. Which is good for the bacteria, but maybe not good for us. So I think of this as the tires. We have 4 tires in our car. That's the bacterial chromosome. Those are the genes that are necessary. You need 4 tires in your car in order for it to move. I know there's some cars that only have 3 tires, but let's just go with the standard car with 4. Now, most cars also have a spare tire. So if you get a flat tire, it's great. You can just change that tire and get on with your way. There are cars that don't have spare tires, and so for them, everyday life is fine, but if you get a flat tire, you have to wait for a tow truck. The tow truck comes and tows you, you get a new tire switched out and everything. A little bit inconvenient, right? But again, day to day, the car still gets you from point A to point B. So plasmids are very much like that spare tire. It's great to have when you need it, but not necessarily need it every single day. Now, when we look at eukaryotic cells, their genetic material is going to be packaged into the nucleus, which is going to be a double lipid bilayer. In that, within that nucleus, we're going to have nuclear pores, and that actually allows for trafficking in and out of the nucleus. Okay, and so if we look at this TEM, transmission electron micrograph, I always love that, so I have to show them when I can. You can actually see where you have these nuclear pores present, so these little holes that are showing up, okay? If we look at the chromosomes in a eukaryotic organism, we're going to have linear chromosomes, and generally there's multiple chromosomes. The more evolved, complex the organism, the more chromosomes they're going to have. There are organisms that are haploid and diploid. So diploid's going to have a 2N state, haploid is going to have a single N. And so if we think about fungi, which would be molds, yeast, mushrooms, they can actually have an organism that has both haploids and diploids, and they actually have triploids, so they have really unique genetic combinations that are present. For eukaryotic organisms, their DNA is going to be packaged and condensed around proteins known as histones, and they also have a more a complex replication that happens because they can use mitosis or meiosis. So next we're going to look at the ribosome. So the ribosome is going to be made up of both RNA and protein. So you may remember from an intro bio class, you're going to have transfer RNA that's going to come in to these binding sites that are present, and then that's going to allow you to add an amino acid on. At the same time, the ribosome is going to be binding to message RNA or RNA to read the code to know which transfer RNA comes in. Within these subunits, we also have ribosomal RNA that's present, and that ribosomal RNA is binding to protein. So if we were to actually denature and dissociate these, we could actually isolate the ribosomal RNA, and each bacteria has unique ribosomal RNA, and that's actually one of the ways in which some of the identification of different bacteria are actually being done nowadays, is looking at the ribosomal RNA to indicate what genus and even species is present. Okay, so just to kind of link this on what's going on currently for identification of microbes. So there's a large subunit and a small subunit that comes together to create the ribosome. And so these subunits are different sizes in prokaryotes versus eukaryotes. And so what we're looking at here in this figure is actually a prokaryotic ribosome. So the large subunit is 50S and the small subunit is 30S. When those come together, they actually bind a little bit tighter, and so they actually have a combined S unit weight of 70S. So prokaryotic ribosome again would be 70S. Now for the eukaryotic ribosome, the large subunit is 60S and the small subunit is 40S. And when they come together, it's not 100, it's 80. And so a eukaryotic ribosome would be 80S. So if we purified ribosomes, we could actually look at the size of the ribosome to indicate whether we have a prokaryotic organism or a eukaryotic organism. And so there are bacteria that previously were identified as being eukaryotic because they could undergo photosynthesis, but based on their ribosomes and also looking at other genes, they are prokaryotic. So the cyanobacteria is an example of a bacteria that's been reclassified based off of our understanding of the differences between prokaryotes and eukaryotes. It can undergo photosynthesis, so again, it used to be called algae, and so you'd hear about these algae blooms over the summer in lakes and ponds, and they're actually usually cyanobacteria. And so nowadays a lot of that terminology is being updated, but when I was back in school, when I was an undergrad, we didn't know cyanobacteria were bacteria. We thought they were algae. And so when I started teaching, I'm like, what happened to the algae, the blue-green algae? I'm like, no, they're cyanobacteria. And again, one way to tell that they actually are bacteria is because of the size of the ribosome. Okay. All right, and next we're going to look at the cytoskeleton. So with prokaryotic cytoskeleton, it used to be believed that they didn't have it, but they actually do. They have these long polymers of actin that act as a kind of rudimentary cytoskeleton. So it does contribute to the cell shape, but we still give the cell wall, that's the major function with the cell wall. With eukaryotic cytoskeleton, it's much more involved. So we have the actin that's present, but we can also have microtubules that are made up of tubulin, intermediate filaments, and microfilaments. So all, as you can see in the figure, all of these different structures come together to provide the shape of a eukaryotic cell. They also can rearrange, just like the actin can rearrange in a prokaryotic and allow for motility. And so something like a amoeba that's a protozoan, you can see this ability of changing its shape, rearranging those actin filaments to allow for that to occur. So next we're going to look at some of the special prokaryotic structures that are present that really make some of these bacteria really special and unique. So because bacteria, they don't have vesicles because they don't have inner membranes. They do have inclusion bodies, so they look kind of like vesicles, but again, there's no membrane on them. So they actually just kind of aggregate different types of nutrients together, and they can store it there. So we can have phosphate or carbon or iron storage. So these magnetosomes are really cool because you can see these almost, it looks like little vertebrates, but these little clusters of iron, and it actually creates a magnetic polarization of the bacteria. And the bacteria with these magnetosomes, a lot of times they'll line up with the magnetic field of the Earth, and it's just, that environment is just, the oxygen levels are right, everything's perfect for them. So again, it's a neat little trick that they use this iron, these little magnets, in order to do that. So if you want to isolate them, one of the things you can do is to take, usually they're found in more aquatic environments, so take some soil from like the bottom of a river, make sure you add some water, and then put a stir bar in, and then just slowly turn the stir bar, because what they'll do is they'll actually attach to the stir bar and try to line themselves up with it. And then you can actually then isolate them that way. We can also have granules. This is a special type of inclusion body that has inorganic compounds found in it. Again, there's no membranes present. So for some of the photosynthetic bacteria, they can have sulfur granules present, but we can have other variations also. Another unique cell structure would be an endosome. So there's certain bacteria that produce endosomes. So bacteria that are found in the genus Bacillus, Clostridium, and Sporosinum have the ability to produce endospores. So some Bacillus, Clostridium, those might like go, oh, I've heard of those. Bacillus anthrax and Clostridium, so they produce toxins also, but they can also produce endospores, which makes it so that they can, they're almost like, the endospore is almost like a seed, so it allows for them to withstand really extreme temperatures and environments, and then they can germinate and produce new bacteria. So in this figure, you can see where we have our endospore labeled. The rest of that bacteria cell would be known as a vegetative cell. So this cell is metabolically active, undergoing cellular respiration and other biochemical reactions. In that endospore, this is where the genetic information is being packaged away. Essential enzymes would be packaged in there too. And so if that vegetative cell dies, because maybe we cleaned it with a chemical or something, it leaves the endospore behind. And again, it could germinate later on. So, I was once teaching the class, and the student was like, yeah, oh, they mentioned about endospores in clinical, and they said that it could survive like, a day on a surface. And next slide. I was like, yeah, well, they've actually been able to take endospores, and they're viable even through carbon dating 250 million years. So a couple days is not so impressive to me. So again, these are very hardy. There's a strain of bacteria, Bacillus subtilis, and so NASA has actually sent it up for many space missions. And one of the things they're looking at is how much UV exposure will actually kill it so that the endospores are killed. Okay, so again, many of these are very resistant to some of the extreme environments, and it just allows for the bacteria to be able to survive, its genetic information to be able to survive. And so what happens is that we'll have this vegetative cell, and there'll be some kind of signaling that happens, whether this is a decrease in nutrients, decrease in water concentration, some kind of stressor that then causes sporulation to happen. And so the chromosome is going to be duplicated, and then it's put into a separate area known as the forespore. And then you end up getting this coating that gets put around it. So we end up developing a cortex on it. So then we eventually have a mature endospore. So when the vegetative cell dies, it's left behind. Once there's signals that nutrients has returned, that the environment is favorable, we then get germination, and so we'll then get a vegetative cell appear. So you can do endospore staining, so you can actually stain for these endospores and see them under the microscope, and they in some cases, just if you do a gram stain, you can see them, and they look like little hole punches inside the cell. So again, there's only certain bacteria that are able to produce this. They're all gram positive, and they tend to be soil bacteria, because if we think about the soil, there's going to be times, there's the winter. It's got these bacteria that live in the soil need to be able to survive these extreme environments, or maybe it's really hot during the summer, so it's no longer favorable. So again, what happens is there's this break in dormancy, and so this could be that there's the presence of water or some kind of specific germination agent, and then quickly it will germinate. And so you can go from having endospores to having vegetative cells within an hour and a half, and for some cells, some bacteria, they have a 20 to 30 minute replication time. So you can very quickly go from having no vegetative cells, no metabolically active cells, to having quite a number of them that they can cause issues. And again, I think of the analogy to like a plant seed. Like a plant's going to produce seed so that it can survive the winter. And again, the winter for a bacteria could be a couple days until it finds a new host, or it could be years, millions of years before it germinates. As I mentioned, you've probably heard of the name of some of these just because some of the toxins that they produce. So Bacillus anthrax, known for the anthrax toxin, but also produces endospores. And then we have a number of Clostridium, like Clostridium tetani or Clostridium botulinum, and they both produce toxins. Okay, and again, traditional cleaning methods, they're resistant to. So medically, and we'll talk about this later on when we talk about physical control of microbial growth, especially in like a medical setting, this becomes a concern. So there's a number of different cleaning methods that are being looked at, including like UV robots that go into the room, hydrogen peroxide sprays, metal surfaces, as far as ways in order to prevent these endospores from being present, so that a new patient comes into the room and doesn't actually acquire an infection, a Clostridium infection. There are some special eukaryotic structures, and maybe not so special. You've probably heard of them before, unlike the prokaryotic structures. So for the eukaryotic structures, we have the endoplasmic reticulum, or the ER. So the smooth ER does not have the ribosomes present, so it has a smooth appearance, and this is where we have lipid metabolism and also detoxification reactions. The more commonly known ER is the rough ER, which has ribosomes docked, where we have protein synthesis occurring, so that the proteins, as they're synthesized, go directly into the ER, where they can be folded, where they can have post-translational modifications happening, and then secretion out to other areas in the cell. We then have the mitochondria, which is commonly referred to as the powerhouse of the cell. Please do not put that on the exam. It makes me sad. It is the site of respiration, the site of ATP synthesis. This is one of the structures that's involved in the endosymbiotic theory. So it's believed that the mitochondria actually originates as a bacteria. It has a very bacteria appearance to it. It actually has its own genetic material that is double-stranded in a circle, very much like a bacteria chromosome. It also has ribosomes that are 70S, not 80S like eukaryotic, so that would be very bacteria. And it's also able to produce energy ATP across its membrane, very bacteria. Right, and so all this evidence supports the endosymbiotic theory that an early eukaryotic cell engulfed a bacteria and then created a symbiotic relationship with it. The bacteria, grew and divided inside of it, so we had multiple copies of that bacteria, of that mitochondria inside, and that gave that early eukaryotic cell an added fitness, the ability to survive because it had something producing ATP for it. That early eukaryotic cell probably did not produce sufficient enough ATP across its membrane. Maybe it lost its ability, it had mutations to those genes that allowed it to do that. So again, just be aware of the endosymbiotic theory. Another organelle that originates through the endosymbiotic theory would be the chloroplast. So again, they have many of the hallmarks or evidence that we see in the mitochondria. It has its own genetic information, a circular double-stranded DNA, just like a bacterial chromosome. It also has 70S ribosomes, a double membrane. And so this is the site where we have photosynthesis occurring. And so we now know that there are bacteria that can undergo photosynthesis. And so it's believed that an early eukaryotic cell that engulfed a cyanobacteria that can undergo photosynthesis. It grew and divided inside. It gave that early eukaryotic cell an added fitness, ability to survive because it had a sugar source it was producing through photosynthesis. And so this allowed the algae to survive, and then we eventually get plants. Okay. Again, plants are covered in botany. Not that I don't care about plants. Another structure would be the Golgi apparatus, and so the Golgi apparatus is important in protein trafficking. So import, processing, and then exporting, and so it looks very much like a stack of pancakes underneath the microscope. And then we have vesicles moving from the ER that are going to bind to the Golgi apparatus, and they use snare proteins in order to do that. And then as the proteins move through the Golgi apparatus, we're going to get modifications happening to it. And then as the vesicles bud off, they can then move to the surface of the cell where they can undergo exocytosis, releasing that proteins that were produced. And then lastly, in eukaryotic cells, we do have vesicles and vacuoles. These are membrane bound. And so this allows for storage of water, food, enzymes, and waste. Vacuoles can be varied. Vesicles can be varied in their size and their function. So for some of our protozoans, we can see these central vacuoles that are really important in maintaining turgor pressure so that the cell stays kind of inflated. We can also have contractile vacuoles that allow for the elimination of water. There's different schools of thinking. Contractile vacuoles potentially play a role in motility because in the elimination of water that can actually drive the protozoan forward and moving. The other school of thinking is that these contractile vacuoles, they're eliminating the water and the function is to regulate the osmotic pressure, and that movement isn't the function of that. So that is for a different type of class to argue which is the true function of contractile vacuole. One of the vesicles that we will talk about are lysosomes. So lysosomes contain digestive enzymes, really important for immune cells. So when we talk about the immune response to microbes, we will talk about lysosomes and how those digestive enzymes actually break down and digest pathogens. And we'll also talk about how pathogens actually have ways in which to stop those digestive enzymes from digesting them. So always remember that microbes are trying to survive, and we are too, and so what's good for a microbe may not be good for us, and what's good for us may not be good for a microbe. Okay, so that concludes the second part for this lecture on cell structures that are found in microbes. Again, the focus is really when you're thinking about these, what structures are found in prokaryotes, what structures are found in eukaryotes, and how do they differ between the two? So what's unique about them? So we really didn't talk much about the viruses. So a little later on in the semester when we talk about viruses, I will make sure I incorporate some of that information also. All right, thank you.